Hello everybody and welcome to Mr. Saucedo's YouTube videos. Today we're going to be going over our third lesson, which is all about DNA replication. So the next three lessons, this one, the next one, for lesson 4, and then lesson 5, they are all very much like the important part of this unit. And so we're going to start with how DNA replicates, how you can make a copy of your DNA. So first things first, let's talk a little bit about what DNA replication is. So you'll recall that when we were talking about mitosis, we said that in a particular phase of mitosis, the replication process occurs, right? And so all that means is that before your cell divides, it has to make a copy of your DNA. And so when does that happen? That happens during the S stage of the cell cycle. So remember, you have like G1 or gap 1, then you have the S stage, and then you have G2. So this is kind of like a representation of what we drew when we were talking about mitosis in Unit 5. But we've got gap 1, this is S, so we're talking about replication right here, then G2, and then mitosis occurs. So remember that your cells want to ensure that before it goes through mitosis, that you have a complete and accurate set of DNA. We talked about that there are these, that there's programmed cell death, apoptosis, if the cell thinks that, oh, I don't have a complete set of DNA at this point, or like, like it kind of has a kill switch basically, right? That's what we talked about. So remember though, all of this occurs during normal cellular division, right? So before cellular division, the S stage is when DNA is replicated. So there are 3 steps in DNA replication. Now, the terms for them are what we're defining here, and then we're going to kind of like give you some pictures and talk a little bit about the enzymes that are involved, because the enzymes are important. So the first step is called initiation. And so during initiation, there's an enzyme, it's called helicase. Remember we were talking earlier about like how things that end in ase, how that means it's an enzyme. And then the beginning part kind of tells you what it does. So helicase is going to unwind the double helix of your DNA. And it does that by breaking the hydrogen bonds. So remember, we said that you have A's and T's and C's and G's that are hooked together with these hydrogen bonds, and helicase's job is just to unzip it. So here you have your DNA strand kind of being separated and unzipped in both directions, right? So it's kind of like unzipping a sweater or something, right? Now keep in mind that there's another enzyme called primase. And again, just to reiterate, primase adds primers, are what they're called. So again, just like before, it kind of tells you what it does. And so it adds these little pieces of RNA which help with the next step. Again, we haven't talked about what RNA really is, but that's okay. Okay. Now, what's the second step? The second step is elongation. So during elongation, there's a class of enzymes, they're called DNA polymerases, and what they do is they attach to the primers. And then when they attach to the primers, they basically start pairing everything up according to Chargaff's rule. So, in other words, if it sees an A, it's going to grab a T and pair it up with that. If it sees a G, it's going to pair it up with a C. If it sees an A, it's going to pair it up with a T. If it sees a C, it's going to pair it up with a G. And it just kind of works its way down the DNA strand that has been kind of opened up by helicase. Step 3, termination. Now at the very end, right at the very end of your DNA, right, you have to remove the primers because the primers aren't necessary anymore, right? Like the whole point of the primers was just to like give your DNA polymerase like a target to be able to like attach. And now you should have 2 complete sets of DNA that are identical to one another. And in order to ensure that, DNA polymerase and nuclease proofreads its work. And so that means that it kind of goes over and it makes sure, okay, wait, there was an A here, okay, there's a T. There's a G, there's a C. That doesn't mean that it's perfect. So nuclease and DNA polymerase are not perfect enzymes, right? They're not going to every single time be able to catch every mistake, but it tries to do so to limit the errors that are getting passed on when your cells divide. Okay, so keep in mind, in order to replicate all of your DNA from all of your chromosomes, right? So all 46 of them, only takes a couple of hours. Doesn't take that long for it to happen. In addition to that, though, the two strands that you produce, right? So like when you are making a copy, one original strand, which is called the template, stays, and then you make one new strand which complements it, and that is called, again, the complement. And so in reality, what you have kind of simultaneously happening, right? So here's helicase splitting apart the hydrogen bonds. You have a DNA polymerase that is essentially just making a new strand as it goes. And then you have another DNA polymerase that is basically doing the exact same thing. Okay? So you have one that's kind of traveling up towards the fork, and then another that is traveling away from the fork. So, DNA replication is considered a semi-conservative process because every single strand of DNA contains one piece of original and then one complement, okay? And so here, if this is our, phrase that's gonna be like replicated, right? This color and this color, if this was like my original entire strand that I made a copy of here, and this was the complement completely, this would not be semi-conservative because it contains an entirely new strand and an original strand. So really when we say semi-conservative, what we mean is that this is the way that it looks. So it doesn't look like this. If this is our secret phrase that we're trying to like make a copy of, half of it is going to be original and half of the other strand is going to be original. And then we're making a half that matches, and that's going to be our complement, and this half that matches is going to be the complement. So in reality, really what's going on here is a semi-conservative process. Again, this would be a completely conservative process where you keep one entire double helix that is the same, and then one entirely new double helix that is the complement to it. But the way that our system works in our cells is we have one half that is original and one half that is the complement. All right, next up. You don't really need to know what like prime means because this is just regular biology, but keep in mind that there is like a direction to the way that we read DNA and the way that your enzymes read DNA. And so the complement, right? Remember, we have that fork. That complement that is facing, this is read 5' to 3', by the way, like that's the way you read this. It's constructed continuously. So it can work, the DNA polymerase that is building it can just run straight through and make a copy, right? Like it can just kind of immediately make a copy. And so this is kind of what you're seeing at the top of this diagram here, right? So we have our helicase again, we have our little RNA primer, and then this kind of yellowy orange looking thing, that is our DNA polymerase, and it is able just to constantly, and so you see this red streak here, this is our complement, it is able to just constantly make a new strand like this. On the other hand, the other strand, which is going the opposite direction, so it's 3' to 5', again, you can just picture it like the direction that you read, whether you're reading from left to right or you're reading from right to left. The DNA polymerase that is doing this can't really work in a straight line. It's reading constantly the DNA backwards. So it's building it discontinuously. So notice it's building a little bit and then it has to stop. It's building a little bit and then it has to stop. It's building a little bit and then it has to stop. So in reality, the one that's moving the fastest, if you will, is the one that is being created from 5 prime to 3 prime. This would be the equivalent to us reading from left to right. On the other hand, if I gave you text and I said, all right, like this is flipped backwards, right? So now you need to read it. It's probably going to take you a lot longer, and that's the reason why this DNA polymerase, it's taking longer for it to read. Now, all these little fragments that it's making, so it's making like a chunk, and then it's making another chunk, and then it's making another chunk, those are called Okazaki fragments. Like that's the term that we use for them. And so the way I always picture it is like, can you read this? This is what DNA polymerase is doing when it's building its complement from 3 prime to 5 prime here, right? And maybe you can, maybe you can't. But what if instead we broke it up into chunks? These would be like Okazaki fragments, right? So it makes it a little bit easier. And I didn't break it up by word. I broke it up like every 5 or 4 or 3 kind of letters here. There wasn't really a pattern to it. And so what does this say, right? Well, there was a cat that sat on a mat. That's the secret message here. Obviously, if you are reading it just from left to right and not flipped around backwards, it's a lot easier to read, which is why DNA polymerase, when it's building its complement 5 prime to 3 prime, it's a lot easier for it to read it. Okay, so here's our picture again. We've got helicase, we've got our template strand, and we've got our new strand being made here. And then we've got our Okazaki kind of fragments being made here on the bottom. Keep in mind that DNA polymerase, once it gets to like the end of the DNA, it really can't read the end of the DNA. And so in case you forgot, those are called telomeres. So they're called telomeres. And in case you also forgot from the unit that we did on mitosis, you lose a little bit of DNA every time your cells divide. And so don't forget, right, that as a result of that, we do lose little bits of our DNA. But that's why our telomeres exist. And remember, telomeres are just pieces of junk DNA at the very ends. All right, so here is how quick, by the way, this process is. So like a machine, this is just a representation of some of what DNA polymerase is doing during DNA replication. So it is reading things very, very, very quickly.
Replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
All right, so again, there were a lot more enzymes than you actually need to know in this little graphic simulation, but you get the idea, right? So if you have any questions, please let me know. But before we move on to the next little part, you might want to go back and then just kind of like read through the process again. So now we're going to practice this, and this is like where we're going to end. So if you're given the following template, so let's say this is a piece of DNA, and I want to write the complementary strand for it. What am I going to do? Well, remember, we follow Chargaff's rule. So A goes with what, and C goes with what. So A goes with T, C goes with G, G goes with C, and you continue, right? So you just kind of work your way through A, C, T, G, G, etc., right? And so just like DNA polymerase, you just replicated DNA. Now, if you were to be doing this completely backwards, kind of with your hand tied behind your back, you'd be doing it like the Okazaki fragment kind of way, right? Making little segments as you go. But you get the idea. Again, if you have any particular questions, please make sure to let me know. This diagram is very kind of important. It does contain some enzymes that we don't care about, like DNA ligase. We don't care about it. But I do want you to, at the very least, kind of like fill it in because it is in your notes. So remember, these little guys are RNA primers. Pick a color. Again, this diagram is green, but pick a color so that you are able to color it in if possible, so that you know that like, okay, that's an RNA primer. Oh, these are also RNA primers, right? We've got ourselves helicase. It is unzipping the hydrogen bonds in the DNA, and so this is also helicase, right? This entire little segment here is an Okazaki fragment. And then these yellow things, which you should also probably highlight somehow, are the DNA polymerases, right? So this is a DNA polymerase, and these are DNA polymerases. Okay, that should be it. If you have any questions, please make sure to let me know.